Hello everyone, this is Masamali and this is my progress report of my first semester. I am the student of Life Sciences for the master program, and the name of my advisor is respected director and Professor Rong Kong Sai, and the name of my co-advisor is respected Professor Shi. So I came to the university on 5 November 2021 after quarantine. On 10 November, I joined the eye research lab in a Tzu Chi Hospital, and on 11 November, from 11 November, I started to learn the basic techniques about cell culture, and onward from 5th December, I started practicing culturing the mesenchymal stem cells. So this is the name of the courses which I was taken in my first semester. The name of these courses is Research Ethic and Education, which I successfully passed and got 91% numbers. And the second is seminar. I attended all the seminar classes and present my own seminar class, and I got 86% marks in this subject. So below from the courses, this is a benchmark which I learned in my that duration. I learned the cell culture technique and extraction of PBMC from a blood and making up condition medium to the cells, and about also know the macroscopy, and also I have idea and knowledge and know about microtome. So this is my progress report. Nowadays I am culturing them in camera stem cells. So this is the record of my cells which I am growing in a cell culture room. So you can check the pictures. You can easily identify all of the cell size and health. That on the D1 the cell size is a little bit, not much more, on D3 and D12, on D7 and D15. So the mesenchymal stem cells needed subculturing approximately on 15, the day of 15. So we should care our cells every day, and we will check it every day about whether it got or it gets contamination or not. So it's so necessary point about the culturing of mesenchymal stem cells. So the aim of study, I am doing culturing the mesenchymal stem cells. The aim of study is that one, that nowadays we can culture stem cells for the researcher and doctors, and they hope that stem cell studies can help to generate healthy cell to replace diseased cells. So when we grow that type of these cells, mesenchymal stem cells, we will provide that type of cells to the doctors, and they will apply a clinical trial, and they will do the drug screening. And also we know about that nowadays stem cell have a much more role in the field of medical. So this is one of the successful report which I got in the start of January 2022, that I collected a PBMC from a Stargardt disease patient blood and induced into iPSC. So I did the process of immunocytochemistry. I identified by the process, by the technique of immunocytochemistry, and I added the different fluorescent marker, and I checked that the colony express so strong. So it will be produced ahead, and I will easily grow and culture the colony of iPSCs. So my future study I will explain now. As you know that the my project name is the development of Stargardt disease specific iPSCs derived retinal organoid drug screening platform. So there is a one female patient of 33-year-old have a Stargardt disease and defect in a gene promo1 at a Buddhist Tzu Chi hospital. I will get their blood, and after that I will apply the protocol to get PBMC. So this is a picture which I edit here in my slides for clearly know how we can get the PBMC from a simple blood, from a normal blood. So this is a material and method that I will apply for my future studies. When I will get PBMC from a blood, after that I will put that PBMC in a condition medium, and I will change the medium every 48 hours, and after that I will subcultured once it's got or it's get 75% confluency. We know about how when it get confluency we can check our media. So after that we will also placed it on minus 80 degrees centigrade for overnight and transfer to liquid nitrogen for storage. So this is the generation of feeder-free human iPSCs from PBMC. So this is a secondary programming kit which we use for the induction of iPSCs. So we can do it after this process when we will place it for a while for a little bit time in a media. So this is a study design which will be take up to 42 days. So it depends upon the condition, because for getting the iPSCs there are three stages. So it depends upon the stages whether the first stage will get completed on the time. So the second stage will be start, and then third stage will be start. So this is the study design of my future studies. And this is a genomic stability of HIPSCs. As you know about that, I am working and I will work on a project which is a genetic disorder Stargardt disease. So I will be check my that genomic stability in a human induced pluripotent stem cells. So we also have in a Tzu Chi hospital we have a cytogenetic lab. We will provide our HIPSCs for check it for a karyotyping. So after that, in vitro differentiation of human iPSCs into embryonic bodies. So after that, HIPSC colony will dissociate with 0.5 EDTA into a small clump. So after that, we will add in a 6-well plate. So the embryonic body like aggregate will maintain for different time points like day 7, day 14 or day 28. So after that we will grow in a incubator at a 37 degrees centigrades with have a 5 degree of carbon dioxide incubator. So after that process it started a in vitro differentiation of HIPSC's early stage to 3D retinal organoid. From this stage now it will be get to early stage of 3D retinal organoid. This step is dependent upon the function of embryonic bodies when it will get functional and will appear to form 3D retinal organoid, because of the cell which got from PBMC is not of a normal human body. That was the patient of a genetic disorder. Maybe it will take up to 3 months to form the shape, or 3 to 4 months it will take. So after that there is a we will also provide, after when we get our human HIPSC retinal organoid, we will submit it to the electron microscope laboratory at Tzu Chi University for ultrastructural analysis using their standard protocol. So after that, this is the expected result which I will be get from my future studies. The expected result, as you know, and you may see that this picture that on a week 4 it will be shaped like that one. It is a picture of a retinal organoid structure, and B, you can check that it is a retinal organoid picture of a week number 6, and after you can check the last picture on week number 8. I wish that and also I hope that I will get like that retinal organoid structure, because in getting the iPSCs induced pluripotent stem cells, we must focus only on the shape, whether it will get the shape or not. And this is, and after that when we will get successfully the shape of our retinal organoid, so after that we will doing a drug screening. We will doing a drug screening, like we will check now our cells whether it will be effective or no. So we can use a CRISPR Cas9 technology, that we will be remove the defect area of a gene, and we will add a we will we will add here the healthy gene, and maybe we can do a clinical trial at the last. So this is my progress report and my future studies, that what I will be do in a future. Thank you very much to all of the OIA members and all of the professors and my respected uh, Professor Dr. Rong Kong Sai and my respected co-advisor Professor Shi. I am very thankful to all of you for expressing my progress report and about my future studies. Thank you so much to all of you. Bundle of thanks. Bye bye.